Hello, and welcome to everyone joining us for today's Select Science webinar titled Improving Spectral Flow Cytometry Workflows with Sony Spectral Systems. My name is Matilde Marques. I'll be moderating today's presentation, and I'm delighted to be joined by our guest speaker, Ben Janusek, Field Application Scientist at Sony Biotechnology. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the right of Auditorium and click to open the Q&A window. During the presentation today, we will also be running a few poll questions. Please keep an eye on for a polls icon. If you could please submit an answer when prompted, it would be greatly appreciated. So, without further delay, I'm delighted to hand over to our speaker, Ben. I would like to thank him for presenting to us today.
Thank you for that introduction. Thank you everyone for taking the time to join us today. I'm going to be talking to you all about standardization mode, the SFA Life Sciences Cloud Platform, and the new robot arm automation upgrade on the ID7000. So we're going to be talking about improving spectral flow cytometry workflows with Sony Spectral Systems. First, a quick introduction to all of the instruments that Sony offers that we have in our portfolio. Today, we're going to be talking about the ID7000. So it's a high parameter spectral cell analyzer. You can get it with up to 7 lasers and 186 detectors. We're also coming out with a new FP7000. So this is our spectral cell sorter. It has the same optical layout as the ID7000. So you can have any panel that you have on your ID will work on your FP. And we have guided software workflows to help you with that sorting. We also have our MA and SH platforms. So the SH is the first one we came out with. It has an automated calibration and really easy guided workflows to help you through your sorting. It's up to 6 colors. And then the MA is the same software, same layout, but you can use up to 12 colors in your panels and 4-way sorting into your 5mm fax tubes. We also have the CGX10. It's GMP ready at the 3 levels of instrument, software, and consumables. And it's a fully closed microfluidic sorter for gene and therapy, cell and gene therapy manufacturing workflows. We also have reagents. So our antibodies, over 12,000 reagent choices for those, and consumables to support all of these platforms. So a quick introduction to the ID7000. As I said before, we have up to 7 lasers and 186 detectors that you can get on the ID. You can use just about any fluorochrome that's out there on this instrument. We have really streamlined, easy workflows. So we're going to be talking today about the standardization and a little bit about the spectral reference library. That makes your workflow a lot easier and a lot more flexible. So a lot more choices in how you go about your workflows. We also have the auto sampler that is included with every configuration of the ID. That enables true walkaway operation, and there's a lot of features there that we'll go over that really do a great job of handling your samples. We also have autofluorescence management, as this is a spectral analyzer. So we're treating autofluorescence just like any other color. And we'll talk a little bit about how that really improves the resolution in your data a little bit later in the presentation. So here's the optical layout of the ID7000. You can see each of the spatially separated lasers over there on the left, and then each of the detector decks coming off of those lasers over here. So this, we have detectors about every 10 nanometers throughout most of the region of the spectrum. And this is going to improve the ability to discriminate between overlapping fluorochromes. This is also going to allow you to move your panels from just about any conventional system. And that process is going to be really smooth and easy to do. This also improves the flexibility for future fluorochromes that are coming out. So we don't need to put in new bandpass filters or anything like that. Because we have these detector decks, we can fit in just about any fluorochrome that's going to come out in the future. And something that makes us unique is our deep UV 320 laser. That's going to further increase your panel design flexibility. We'll go over a little more detail about how that works a little bit later in the presentation. Here are some examples of some colors that you can run together on the ID that you can't on a lot of conventional systems. So we have some examples off of the violet laser here, off of the yellow-green laser, and off of the red laser. And what this is going to allow you to do is to combine panels that you previously may have had to split up if you're using conventional systems, or expand your panels so you can get more information out of your samples. And so here's some more detail about the 320 nanometer deep UV laser. And so we talked a little bit earlier about how it's going to increase your panel design flexibility. This is because a lot of colors have secondary excitation by the deep UV laser. And this extra data is going to allow us to better separate out those colors and improve resolution. Autofluorescence is included in this. So a lot of the autofluorescence in your cells comes off of the 320. And so that increased data is also going to allow us to improve the resolution. So we can see down here some data from this paper that without the 320 nanometer laser included, you can see the resolution is a little bit tricky on some of these markers. But when we include it, we get much better resolution, and so you can better see your populations. So how do we set up our standardization mode? This is going to be a one-click feature, and it's enabled through our daily QC. In our daily QC, you're going to run what are called the line check beads. They're going to be used to set up the instrument. And one of the things that happens is that the line check beads are going to be corrected back to the same standard every day. So this is going to allow you to maintain the consistent output of the ID7000 for longitudinal studies. Another thing that happens during the during the standardization mode set up during the daily QC is that the ability to run your single color controls and your fully stained samples at different voltages is enabled. And this is because all of the data that is recorded on the ID is corrected back to the same internal standard between the different detection decks. And we'll talk a little bit later about why this may be important for your data, but this is really going to increase your workflow flexibility. So you have stability and flexibility coming out of your standardization mode. So we'll start with the stability of the instrument. So here's some data of 8 peak beads run at 10 different time points over 175 days in the PE region. And you can see the stability of each of those peaks. The CVs of each of these are under 5%. And so when you're running longitudinal studies, you can be confident that each time point that you run at, you're going to be getting a consistent output from your ID. We have a couple examples of some different studies with some actual data that were enabled using the standardization mode. The first is this paper out of a group from Denmark, studying patients with MS treated with anti-CD20 therapy. And so they had an 8-month study. There were 15 patients, and there were 3 follow-up time points. And because they were using standardization mode on their ID7000, they were able to use the same standardization voltages and be able to compare all of the data from each of these time points. And this allowed them to observe some really cool changes in some NK and CD8 cells over the course of this study. We also have a webinar by one of our users, Paul Rouse. He's an R&D scientist and an expert in thymus biology and spectral flow cytometry. And he talked about how he is standardizing his ID7000. And so he talks in this webinar about the conversion from our old normal mode and the improvement in the standardization of the output of his ID7000 using the standardization mode. He also talks about how standardization mode allows consistent output of his ID7000 over the 10-hour working period after the daily QC is run in the morning. He also talks about when his company purchased a second ID7000, he was able to use standardization mode to provide consistent output within plus or minus 5% limits over time on both of those instruments. So he presents 8 peak data to show all of those. And I recommend that you watch this webinar if you're interested in seeing the data yourself. We talked about the workflow flexibility of the standardization mode. And so when you run your single color controls, we're going to calculate spectral references. And those spectral references can be saved in a library. This will allow you to use previously recorded spectral references on any future panels or experiments that you want to. So you don't have to run single color controls each time that you run your experiment. Obviously, with tandem dyes, those can break down over time, so you want to be careful about how much you reuse those. But there's extra information that you can input on your spectral references when you input them into the library, like an expiration date and stuff like that, to help you keep track of all the spectral references in your library. So I mentioned earlier that you can run your single color controls and your fully stained samples at different voltages. And so what you can do with your single color controls is run them at a preset voltage. So you don't have to worry about setting up your voltages on single color controls. That's a much easier workflow than you might be used to coming from a conventional system. And then when you run your fully stained samples, because all of the data is corrected back to the same internal standard, we can have accurate unmixing when we increase our voltages. And this allows us to increase the stain index and really improve the resolution of your samples. So we have two methods of doing that, called the 1max and the all-max method. So with the 1max method, you're turning up all of the voltages on your detection decks together until you have your peak fluorescence just below the saturation point on one of your detectors. And then on the all-max method, you're turning up all the rest of the voltages on each detector deck where you have a fluorochrome primarily excited by that laser until the peak fluorescence on that deck is just below saturation. And you can see the improvement in the stain indices down here when you go through these two methods. Here's a little bit of data to show what the actual results from this are like. So here is the preset voltage with the beads. And you can see on a lot of these populations how the resolution improves as we turn up the voltage. So this enables users to get the best data possible from your samples and not be limited by the voltages that you run at on your single color controls. So you get better data and better workflow flexibility. So summary of the standardization mode. So we have the stability with the consistent and the unbiased results for your longitudinal studies. And we have the flexibility with your single color controls and the ability to run at different voltage between your single color controls and your fully stained samples. We're now going to be talking about the SFA Life Sciences Cloud Platform. So when you do your analysis on your samples, usually you're going to be doing your manual gating on your bivariate plots. But the problem is, as we start running larger and larger panels using the ID7000, the analysis time is going to increase as the number of colors you're using increases. And anyone who's analyzed a 30 or 40 color panel will know how much time it takes to go through all of that gating and do that analysis. What people have started implementing is these dimension reduction algorithms such as t-SNE and clustering algorithms such as FlowSum. What these allow you to do is to kind of boil down all of the differences in your data to a 2D plot and identify larger differences between samples in certain populations, so you can do a more targeted, efficient analysis. So a lot of people are doing these analyses where they're starting with these dimension reduction or clustering algorithms, and then they go back to the bivariate gating. So we launched the ID7000 at Sony in 2020, and many users, as they started running large panels on our instrument, they wanted advanced analysis. So we recognized that there was a need for advanced analysis of ID7000 data. So we came out with the SFA software. That stands for Spectral Flow Analysis, and this is a life sciences cloud platform. So what this allows you to do is take ID7000 data and upload it into a cloud-based platform that allows you to use these algorithms to analyze your data. The user interface of the SFA matches the ID7000 software look and feel, so you don't have to learn a new software when you're using the SFA. We also have a batch learning FlowSum This is improved clustering algorithm that's based on FlowSum and was developed in-house at Sony. And we also have interactive analyses. So it's a connected workflow between your conventional and advanced analysis plots. So you can go back and forth in a really easy and efficient way. This software is ready for every level of user who wants to use these advanced analysis algorithms. And the way that it's set up where you upload your data to the cloud also allows you to share your data with any collaborators that you may have. So the way that this works is you're going to record all of your data on the ID7000 and analyze in the ID7000 software. And then you're going to export your experiment in one file. That file is going to be uploaded to the SFA, and then you upload your data to the cloud. And this is where you're going to do all of your analyses and be able to share data with collaborators. So there's no plugins because we're uploading data to the cloud. The batch learning FlowSim are FITSNI, which is just like a TISNI, and UMAP are installed in the SFA on the cloud analysis backend, and they're accessible to the licensed users. So FlowSum is a novel clustering algorithm that was published in 2015, and it's becoming more and more widely used as people are doing more and more high parameter flow. In the original paper, there was an area of improvement that was identified that the original algorithm was not optimized to run in parallel. And so that was identified as a possible improvement. And so Sony implemented that improvement in our batch learning FlowSum. So we changed our learning method from online learning to batch learning, and principal component analysis and the batch learning were implemented in the native code to enable more efficient analysis using our batch learning FlowSum compared to the original FlowSum algorithm. What this allows you to do is analyze your data in much, much less time. So we have a over 4x reduction in the time that it takes to analyze your events. So we have a 42 color panel, and there were 5 million events, and we were able to really reduce the amount of time it took to analyze that number of events using batch learning FlowSum. When you take a look at all the algorithms, it's not just the batch learning FlowSum where you see a decrease in the amount of time that it takes for analysis. So we're comparing here about 1 million events, 42 colors, and this is a competitor software and the SFA software. And there's about a 3x reduction in the amount of time it takes to perform your entire analysis workflow, including UMAP, FITSNI, and the batch learning FlowSum. We have a publication here that used the SFA platform. So this was a 43 parameter panel on PBMCs. This was a 6 laser ID7000. And there were 41 antibodies used in this panel, along with 1 autofluorescence channel and 1 viability dye. And FITSNI was used to identify 28 subpopulations that matched the primary subsets that were found through bivariate gating. And we were able to identify using FITSNI some really, really rare populations, like this population of HSCs and this progenitor population that were less than 0.01% of the PBMC population. So a summary of the SFA software. Because it's a cloud-based solution, it's a lot faster and it's scalable, and it's ready to use if you're used to the ID7000 software already. It's already pretty user-friendly, so you don't have to use any more time to learn a new software when you're doing these advanced analyses. The batch learning FlowSum, the FITSNI, and the UMAP are included, all ready to go. And the batch learning is an in-house improvement on the original FlowSum algorithm. We have interactive analyses. So if you're using these advanced analyses, you can go back and forth between these and your conventional bivariate plots to interrogate your data and find some interesting results. And then because the data is uploaded to the cloud, you can share that data with any of your collaborators. So now we're going to talk about the automation upgrade with the ID7000 and the new robot arm. So our auto sampler delivers really high performance. There's no hardware changes between using 5 mil tubes in your 24 tube rack, or you can use 96 well plates, U bottom, V bottom, flat bottom, half deep, full deep, 384 well plates. No hardware changes. They all just fit into the auto sampler right here. There's vacuum driven sample introduction, and your probe is going to mix each sample individually, and you have multiple mixing modes. So you have nice flexibility with that. There's also a low dead volume mode. So this probe is going to go down and just lightly touch the bottom of your well or tube and then come up just a little bit. So you get just about everything out of your samples. We also have a batch analysis feature that allows you to export, once you've analyzed your data, all of your statistics in a CSV file for an entire sample group. It also will give you a PDF of the worksheet of each of your samples. We have multiple worksheet modes. You can have a shared worksheet in your sample group where every change you make is shared across the entire sample group, or you can have individual worksheets if you want to do individual analyses for your samples. And when you export FCS files, you can enter up to 10 custom keywords to help you maintain and manage information across different platforms. So the new robot arm comes with the ID 7000 if you purchase it. It enables anyone to locate the position of a plate in the stacks in under a minute per plate. And you can do 96 well plates or 384 well plates, as well as full deep and half deep plates. There's a barcode scanner to support if you have barcoded plates, and you can schedule all of this in a really easy to use software that's drag and drop to quickly and easily create your automated workflows. So we have a random access microplate hotel or stack that enables you to use up to 16 standard height 96 well plates, or you can use 8 deep or half deep plates. And this allows you to retrieve these plates in any order that you program in the software. So when you purchase an ID 7000 with the plate automation capabilities, we're going to build a purpose-built door system. And so an automated door is going to replace the manual door, and it's going to be coordinated by the plate handler communication software to align with the plate input and output cycles. And so the software for the automation support includes a methods development editor, a dynamic scheduler, and a really simple user interface. So you press play and you follow the configured prompts. And like I said before, it's a drag and drop software. So it's really easy to use and operate. So a summary of the new Hudson robot arm. This is what it looks like when it's right next to the ID. So you have the random access plate hotel, and then the robot arm here, and then the new easy access door that will be installed with the ID. So it makes your automated plate handling available for the ID. You do need to modify the door, and this is compatible with a barcode reader on the short side of the plates here. And now be happy to take any questions about any of the information that we've presented today.
Thank you, Ben, for that interesting presentation. I hope that everyone listening enjoyed the webinar and found it informative. We will now move on to the last part of today's webinar, the Q&A session. As a reminder, to ask a question, you just need to simply head to the engagement window to the right of Auditorium and click to open the Q&A window. So, to kick off this final portion of today's event, the first question I have for Ben is, can automated acquisition be paused to fill sheet or empty waste tanks without causing errors?
Yes, the automation can be paused without causing any errors. And the software will actually let you know when that's necessary. It has weight sensors for each of the tanks. So it will let you know if the sheet tank is running out of fluid or if the waste tank is getting full.
Great, thank you. And can multiple users in the lab share an SFA license?
Yes, so when you get an SFA license, you can have up to 5 logins for each license. So if there's multiple users in their lab, they can each have their own individual logins.
Great, thank you. The next question I have says, Can you use a mixture of reference spectra from the Spectral Reference Library and fresh single color controls for experiments?
Yes, you can, and that's actually a really nice workflow flexibility feature of the instrument. And so what you can do is for tandem dyes that may break down, like I mentioned in the presentation earlier, you can rerun those for your experiments if you experience breakdown of those dyes, and you can pull your non-tandem dyes from the library.
Lovely, thank you. And does Sony provide technical support if my lab is interested in running longitudinal studies using standardization mode?
Yes, we do. If you're interested in technical support and working with us to help support you on your longitudinal studies, then you can contact your field application scientist and we can help you with that, with any questions you may have.
Great, I think we have time for one more. So, what is the cloud size limit for SFA?
It's 500 gigabytes for the license.
Lovely, thank you. Um, I'm afraid that's all we have time for today. I'd like to thank Ben for today's discussion and presentation and for this lovely Q&A session. And thank you to everyone joining us online for submitting your questions. We hope that you have found this a worthwhile session. If we didn't quite get to your question today, we'll follow up with answers to these after the webinar. And if you have any other question after today's session, please feel free to email myself at editor@selectscience.net. If you are interested in receiving a certificate of attendance, this will be shared with everyone who has attended live today via email a few days after the event. And for those watching on demand, a certificate can be requested by emailing us at the same email address. And if you'd like to listen again to today's webinar or invite a friend to do so, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.